So for today, sorry about that, I'm going to talk about how the streams in V1 go through to V2 and a little bit higher, just very briefly about that. And then also what's the difference between simple and complex cells, which are in the interblobs. So that's about the first half. And then the second half we'll basically sort of review for the midterm, the main points. So if we head over to this picture here, so what is this picture? So this picture it's a picture of the cortex that has been flattened, and then a thin section has been taken parallel to the cortex, and it's probably going through something like layer 5 or 6, something like that. And so if you look at this, so you can see there's an obvious border right here. That's a very clear border like that, sort of going right along there. And this is about where the star is that I always draw, and here's the circles, like the vertical meridian that I always draw, and here would be the horizontal meridian. That's the horizontal line in the visual field, and then it sort of, this is where that split is occurring in V2. So there's the rest of V2. So this basically this guy is V2, and V2 it goes all the way up like that. That's all V2, and this is V1 over here. And so now you can see the blobs. So you can see it's extremely clear. Like there's a blob, and there's another blob. And so what are the interblobs? Those are just a little lighter area in between. So that light area in between is where all the different orientation selective cells are. And then the dark spots are where there's a lot of cytochrome oxidase, and those are the cells that are seeing brightness and are kind of throwing away orientation. But it turns out when you look in V2, that V2 has some compartments in it too. And so here's one over here. This one is called, you can see it's a little blobby. You can see there's sort of like a little blobby. That one's called a thin stripe. So this guy is thin. And then in between it, there's an interstripe. So this area in here is less, darkly stained. And then you can see this guy, which is a thick stripe. And so you have these little stripes instead of blobs. So this guy is thick. And so I'll talk about how V1 is hooked up to these. But this is an example of the variousness of streams. So like you can't see, if you go back to V1, you can't see layer 4B, because layer 4B is from a different layer than what we're looking at right here. But in V1, the streams were blobs, interblobs, and layer 4B. And in V2, you can see they're different. Okay, so now let's go back. Let's get rid of the overlay. So, okay, there we go. Okay, so let's talk about how the streams go forward from V1. So if we draw that little diagram of V1 again, there was layer 4. That was layer 4, and there's layer 1 up there, and we had some blobs, and there's a blob. So that's a blob. And then inside layer 4, there was this layer, the 4B, that actually didn't even have any input from the geniculate. So that's 4B. So basically it's 4B and then a blob. and then interblob. So those are the those are the three streams coming out. And so when they go to V2, it turns out they form new streams. But how did they kind of like initially figure this out? So if we make a drawing of V2, it's a mirror image of what I was just showing you on the. I had V2 off to the left. Now we got it off to the right. Okay, so there's V2. It's a long strip, and what does it have in it? It has a thick stripe. I'm drawing them a little bigger than they are, just so we can see things. It's got a thick stripe, and then an interstripe, and then a thin stripe, and then another interstripe, and then another thick stripe. So how is this figured out? It was actually figured out originally by going yet one level higher. So if you go up to MT, so MT is a higher area, people did an injection into MT, and what they found was very clear stripes. You could see that the neurons just came out of the thick stripes. So if you inject something here that goes backwards through the axons, and then look where the cells are, you very clearly see these stripes. And then they stained for cytochrome oxidase, and it corresponded to the high cytochrome oxidase area. So that's how this was originally figured out. And then if you look, there's another area, V4. Some people call it different things. But if you look at area V4, that was getting input from the thin stripes, but also the interstripes like that. And so you could see that there were stripes again if you injected V4 and looked. So that's how they figured this out originally. And then they went, back to try to figure out how does V1 hook up to V2. And so the first thing they found is that if you look at 4B, not unexpectedly, it projects to the thick stripes. So this is, thick, inter, and thin. So that sort of makes sense because those guys were going to MT, and it turns out the blobs project to the thin stripes. Let me make one more thin stripe up here so I don't mess things up too much. So the blobs go out and project to the thin stripes, and that sort of makes sense because they're thin, they're sort of blobby, and the interblobs projected to the interstripes. That was the original story. And the original story was simple, like streams are just going through V2, but it was wrong. So, what happened, it turns out that, layer 4B sometimes projects to the interstripes, and the interblobs sometimes project to the thick stripes. So, like I said at the very beginning, you can't trace these streams all the way through. There's a set of streams here, and then they sort of mix them up, and then we get another set of streams here, and then they mix them up some more. So if you look in MT, for instance, there's little patches in MT, and the cells are different in the little interpatches. And if you look in V4, you see the same thing. There's a complicated little patchy system. And so this idea of having a basic map and then having multiple areas, or multiple versions of the information sort of passing through each map at each point is common throughout the nervous system. And on the good side, it's not 20 streams. It's usually like two or three. So it's a reasonable number that you could remember. So that's pretty much just a tiny fragment of, there's a ton of research that's been done trying to sort of figure out how all these things interrelate. But in general, it follows this this idea that we start off with the retina, and then the retina does a bunch of things and makes these streams, out of one source of information. And so we have, magno stream, the magno cells in the retina, and then there's smaller cells in the retina, and then there's these blue cells in the retina, and those go up to the that's one case where the streams sort of don't change, because the LGN is pretty much just a relay, conio. So this is the DLGN here. DLGN, and these are the retinal ganglion cells. So those are cells, in the retina. So there's retinal ganglion cells in DLGN. And so we have three very clear streams, and they go up into the cortex. They kind of get mixed up, and then we end up with three new streams, which are where the blobs, the interblobs, and layer 4B. So that, and then those project up to V2, and then they get mixed again, and then you've got some other streams over here, and then those project up to yet a higher area where they get mixed up again, and there's another like 6 or 7 areas down on the right that we haven't talked about. Now, you might think, and the other way that people tend to think about this is they tend to think about it very serially. So you have, the retina, it goes to the LGN, it goes to V1. I forgot to label this. It goes to V1, that goes to V2. But, there's just a little extra connections. And so one example is, here we have, the dorsolateral geniculate. We focus all this attention on the dorsolateral geniculate. But there's another structure down here called the pulvinar. And the pulvinar, funny word, it means pillow. It's the pillow of the thalamus. The pulvinar is a huge thing. It's about 6 times bigger than the geniculate. 6 times DLGN. And the pulvinar actually has probably 6 maps within it. So it's kind of like 6 additional geniculate-like things in the thalamus. The pulvinar is in the thalamus. And so how does that one get input? Well, the retina, projects to the superior colliculus. So the retina projects to the superior colliculus in addition to going to the, actually I should put that up a little higher because it's in between here. Okay, I'll put it here. Let's put it in pink. Okay, superior colliculus. That's in the midbrain. So the retina projects to the superior colliculus, and then the superior colliculus has a big projection to the pulvinar, like that. And then when we look at the pulvinar, it turns out the pulvinar projects to V2. And so the projections from the pulvinar may be the thing that actually define the stripes. So there's projections from the pulvinar directly into the stripes that sort of skip the stream coming from V1. And just for good measure, the pulvinar projects to V1, and V1 projects back down to the pulvinar, and V2 projects to the down to the pulvinar. So it's quite a people study one thing and then they just kind of focus on that and apply the function to that. But the reality is there's a lot more parts, and they're sort of going back and forth, and all of them have streams within them. And there's a bias in the literature. So if you look if you read, you'll find a ton of papers on the lateral geniculate and on V1, and you'll find just a handful of papers on the pulvinar, even though, like I said, it's the pillow of the thalamus, and it's 6 times bigger than the geniculate, but it hasn't been studied 6 times as much. It's probably been studied about 1/20th as much. And so that's an interesting thing to sort of think about when you're reading things, because there's kind of a background assumption that the geniculate must be really important, or V1 must be really important. But there's these other pathways that sort of skip. So, yes? Yeah, so pulvinar could be one of the, so the question was, is this related to blind sight? Yeah, so there's some evidence that some of this information, like if you damage the geniculate or damage V1, you can still see things, even though you don't have sort of a conscious, you often don't have a conscious percept of seeing it, but if somebody throws something at you, you can actually grab it. So, or if you just put something up, say, look at this, and the person say, I can't see it, and then they'll look at it. So, yeah, so V1 seems to really be necessary for this sort of bright, conscious experience. You can't, just by sort of activating your higher areas, if you don't have a V1, you can't actually sort of get this feeling of like, I saw something, I saw like a bright, conscious experience. So V1 seems to be required for that. So yeah, so there's lots of interesting areas here. The pulvinar is also heavily involved in attention, like what you're attending to. It's hooked up strongly to parietal cortex. So, okay, so that was V1 going to V2, and so now I'll just leave that up for the time being. So now let's talk about simple and complex. And so now we're talking about the properties of the cells just in the interblobs. So this is, so now we're just talking about these guys that are sensitive to different orientations as you move across the cortex. So they're divided, into at least simple and complex. And so what does that mean? And so put simple, complex, and then there's some cells called hypercomplex. I always make fun of that because they're moderately more complex, not really very complex. Hypercomplex. And so what we want to do now is just talk about kind of the actual mechanics of how these cells code orientation. And so to do that, we have to talk about, so if we look, here's a simple cell, and this was originally defined by Hubel and Wiesel, and so why is it simple? So first we have to talk about the receptive field. So remember, the receptive field is the region in the world, if you fix the eye, it's the region in the world that when you put a little visual thing there, the neuron responds. So that's the receptive field of a neuron. So that receptive field, so this guy is the receptive field. Receptive field. That guy is actually quite a small little region of the world. So how small is it? Well, if we're talking about a simple cell in V1, so simple cell, so put V1 in here, V1. So a simple cell in V1 interblob, if you pick one from the center, near the center of gaze, its whole, width of the receptive field, so like this width in this direction, might be something like 1/5 of a degree. So now, what is a degree? So 1/5 of a degree of visual angle. And so what is visual? Visual angle is a way of sort of saying how big something is on the retina. And all it means is, here's your eye, and you're looking out at the world, you're looking out at something in the world, so here's something out in the world. It's just, basically, if we just take this angle right here, like what is that angle? And so that's a way of defining how big it's going to be on your retina. It doesn't depend on how big the object is or where the object is, because if a smaller object was closer, it could have the same visual angle. So it's just it's an independent way of saying, like, how big is the object going to be on your retina? That's what the meaning of visual angle is. And so this is kind of a little unfamiliar thing to think about, but what is kind of like some handy things about visual angle? Like the moon is about 1/2 degree. So if you look up in the sky at the moon, it's always about 1/2 degree of visual angle. So that means that these receptive fields are smaller than the moon. So there'll be multiple receptive fields in V1 going across the moon. So V1 is a strange area. It's looking at the world through these teeny tiny little straws. It doesn't really care too much about what's going on around. It's just looking at a tiny little thing, like what's in the upper left part of the moon. Another simple way of visual angle is that your thumbnail at sort of arm's distance is on the order of a degree, something like that. You can compare that to the moon, like put it up to the moon and see how big it is. You might have a smaller or bigger thumb. So that's a visual angle, and so here's this receptive field. It's the part of the visual field that, if we're recording from some neuron here in the interblobs, that essentially causes the cell to become active. So what Huebel and Wiesel immediately figured out was that if you look inside the receptive field with a really, really tiny dot, what you can find is that there are subregions in there, inside that receptive field, like this. I'll put 3 in there. Sometimes there could be more, but say there's 3 subregions in there, and those subregions either respond to off or on. So remember that off is not inhibition. Off means an excitatory response to dimming, and on means an excitatory response to brightening. And where do those come from? Well, those come back from the retina. Remember, the retina is all off, but then one of the things that you get early in the retina is on versus off. So that's something that is initially generated in the retina, but you can see it up in the cortex. And if you put a little teeny tiny spot, like turn on a teeny tiny spot in the on field, you'll see a little response from the cell. Now, if you turn on a teeny tiny spot in the off field, it doesn't like it, because the off field likes brightening. It likes dimming, sorry. It likes to see something dimming. And so how do you stimulate the off field? You just turn the spot off. So if I turn the spot off, it dimmed that part of the world, and then the cell will respond. So why are these cells simple cells? They're simple cells because you can explain their orientation selectivity on the basis of the arrangement of these on and off fields. And if we look at sort of like an LGN cell might say we have an off-center LGN cell. And here's the inhibitory surround, which is different, like I said, than off. So if we have an off cell center surround, and when we actually sort of this thing is in a DLGN projecting up to V1. If we look at the size of those things, they're about what you would expect. So So here's, if I put that off geniculate cell over the cortical cell, it's about the same size. So it's pretty obvious how this is working, where you're getting little rows of on and off cells that are all hooking up to the same simple cell. And that's what causes the cell to be orientation selective, because now you've got these elongated regions. If you look at the geniculate doesn't have elongated regions, slightly, but almost circular. And so when you go up to V1, you see the receptive fields are bigger than the geniculate, because the geniculate ones are really tiny. So the receptive fields are bigger than, this is DLGN, receptive field. That's what this guy is. So that's a simple cell. So a simple cell, it's simple because we can sort of explain to ourselves why it's orientation selective by looking at the arrangement of the on and off fields. So now when you look at this, and you know, what are we comparing this to? Well, we're comparing this to, say we put a bar right here in the field. We get a good response to that because it's the right orientation. And if we put a bar across this way, the response is very poor. So, you know, bad, good response. So we can test with a good size bar and determine whether this cell is orientation selective. And then we can test with a little tiny spot and then find out, oh, we've got these elongated regions in there. So that's a simple cell. Yeah, a simple cell turned out to be a little more complicated. But let's think about now how this cell responds. So say I've got a bright object here. The response to the cell is really quite complicated. It's not just a straightforward edge detector. So imagine I've got this edge moving in. It's at the right orientation. So it moves in to the, let me get out of the receptive field. Okay, so it moves into the receptive field, and so as it starts to move into this on region, it's brightening the on region. And so the cell would suddenly start firing when you did that. And then when I went past there, the response would drop because now the whole thing is bright, it's not changing. And now I'm brightening up the off field, but the off field doesn't care because the off field only likes it when it gets dimmer. And so there'll be no response as I keep going. And then I would get back into another on field, and suddenly the cell would start to respond again because I'm brightening up the on field. And then now, as I'm leaving, there's the rear view. So now I'm dimming the on field. The on field doesn't care about that because it only likes it when it gets brightening. But then when I do this right there, I'm dimming the off field, and the off field would really like that a lot. Meanwhile, the on field over here is just on the whole time, and so it's not giving a big response because you didn't suddenly brighten it. And so then the off field now gives a big response, and then now I'm dimming the on field, but the on field doesn't care because it only likes brightening. So the bottom line is, it's a mess. That's a terrible mess. That doesn't give you a very good representation of orientation. It's all mixed up with, is it a bright edge or a dark edge? And so that's why we need complex cells. And so what are complex cells? So complex cells they look pretty much like simple cells, except when we test with a little tiny spot, we can't find any subfields. So what happens is if we turn on a little spot here and then turn it off, both of those give us a response. So when we turn it on, that's an on response. When we turn it off, that's an off response. And so you look across the entire receptive field with a tiny spot, and you can't actually see, there's no difference in the response to on and off. But if you use an oriented stimulus, you'll see it will respond if you put a edge here, it will respond if you put a dark edge. So, you know, what's a dark edge? Well, we could just have like a bar that's bright all around it, like a black bar on a white background. This is, you know, a dark bar. So we could try with a dark bar. It turns out a dark bar is okay too. A dark bar will work. If you do the wrong orientation, it won't work. So, you know, no for this, yes for this. But it would also be yes for a dark bar. So here's a dark bar. Imagine we had a dark bar. So it's all light around it. So you can see that this, so yes for this guy. So you can see that this cell is a better edge detector because it won't get messed up by whether the bar is dark or light, and it will just signal the orientation. So this is a better orientation detector. And the question is, how do you make one of these? Well, so this is, let me just put right in here a dark bar. So how do you make one of these? You could make one of these by adding up a bunch of simple cells that were slightly offset in where their on and off regions were, because there's other simple cells. You know, there could be a simple cell with just two. You know, you could have a simple cell that looked like this, that just had a, you know, like an off and an on, something like that. If we blew that one up the same size as this and added it on top, then you could essentially generate something that is a better edge detector. But here's the important take-home point, which is you've lost something. So what have you lost over here? Well, you've lost exactly where the bar is. So you don't know exactly where the bar is anymore. So if I come in with a little with an edge into this thing, it just responds the whole time. And then when it leaves, it responds also. And so you don't know exactly where the edge is anymore. You could put the edge over here, and it responds. If you put the edge over here, it responds. If you make a dark bar over here, it responds. So you don't even know what color the edge is. Now, is that a good thing? Well, yes, it is. If you're trying to sort of figure out the shape of an object, you might want to throw away exactly where the edge is and whether it's a dark or a light edge. Now, somebody else might need to know whether it's a dark or light edge, and in that case, they would have to get information out of the simple cells. But this is kind of like the first example of an aperture problem. And the aperture problem idea is that if we've got these little detectors that have, say, on and off cells, they give you good information, but they're kind of stupid, and they get confused by all different sort of like bright versus dark. Now we've solved the problem, but at some expense. We don't know exactly where the edge is anymore. And we're going to see like 20 examples of this where we slowly compute things about the input all the way sort of losing specific information about where stuff is, exactly where stuff is. Okay, so that was a complex cell. And then Hubel and Wiesel, I always chalk this up. The reason they called it a hypercomplex cell is because they knew it wasn't that complex. It was kind of a sop to the idea that the cortex is doing high-level things, and so it's hypercomplex. And so what is the hypercomplex cell? Hypercomplex cell it's a complex cell, except at the end of it, it's got this region that's inhibitory. It's not off, it's inhibitory. So this is inhibitory end stop region. And so how does this work? So this works if you put a bar into the receptive field like that, and you don't go into that region, the response is good. But if you extend that bar a little bit into the end stop region, then the response goes away. And so what's the best thing that this hypercomplex cell likes? It likes the end of a line. So it likes, if you put something in there that's oriented in the correct orientation, it won't work if you put it this way. So if you put something in that's the correct orientation, you'll get the biggest response if you go all the way up to the end stop regions, but not into it. And so this could be a cell that is, selective for looking for actually detecting when you've got a line coming to an end, because sometimes that's an important visual feature. But it's not just that. So another thing that people have suggested is that this might be involved in detecting curvature. And how would that work? Well, if you say I put a line that's about the right orientation, but it's curved. So it's going to give you a response because it's approximately the right orientation, but if it's curved, it won't go into the end stop region. And if I put one that's not quite as curved like that, it'll go partly into the end stop region. And so basically, it could also, so it could detect something like, terminators, like, line, edge terminator. Say line terminator. That's one thing that we might want to know about. Just like the end of the end of a line. But it could also detect curvature of an edge. And so that's another suggestion that people have made, because if you if you look at, you're trying to detect a little edge going around, around the the curve of my forehead, these kinds of cells are going to give you a signal that varies. So there's a hole here, and this hole is filled by another cell called a simple hypercomplex cell, and that's actually the name for it in the literature. So with this kind of hierarchical thinking, the idea was, you get simple cells, and then simple cells make complex cells, and then you get really hypercomplex cells. But it turns out that some simple cells have end stopping. And so if you look at one of those, say this guy has 4, some of these cells can have 4 regions in it. So here's, say this is an off, on, off, on. But those guys can also have an end stop region. So if you make any of the bars too long and they go into the end stop region, it won't work. So that's a simple hypercomplex cell. So that's just a quick and dirty picture of some of the basic, more details of how the physiology of these orientation selective cells work. And really the take-home message was, you start off with simple cells. They're giving you very specific information about exactly where an edge is. They're orientation selective, but they get confused by is it a bright edge or a dark edge? How wide is the edge? That's going to affect things. When you go to complex cells, they've solved that problem. They can detect an edge. Now they can't tell is it a bright or a dark edge, exactly where the edge is, but we've got something valuable, which is there's an edge there. And so in order to do that, we've kind of thrown out some of the exact information about where the edge is. And we'll see this again and again. Okay, so that was simple and complex. So now the plan is quickly, any questions about V2 or simple and complex? Yeah.
What is the dark bar physically?
Oh yeah, what is the dark? Yeah, I didn't do a good example of it. So imagine this is the whole world, okay? So we could have 2 things. We could have a light bar like that, and maybe we're moving it, say. And then we could have a dark bar. And a dark bar it's just a little bit of darkness with light all around it, like this. So this whole thing is light. It's like a shadow. So there's there's a it's all filled in. It's like a big bright patch of light, but then it's got just one little oriented darkness in it. So it's just the exact opposite. You just turn the light parts off and turn the dark parts on, and then you get a dark bar. At first, you don't think of that as a thing. The visual system sees, there's actually more cells that respond to this situation, this shadow situation, than respond to the light bar on a dark background. And all coming from these, it all comes from this on versus off business that starts in the retina. Okay, so to quickly review the course, and what's on the exam. I haven't made the exam yet, but it's always about the same. So we started off with the Nernst potential. So the first question will always be about the Nernst potential. So to sort of get a concrete idea of how it works, and it has to do with the concentration ratio. So that's what the Nernst equation is. It's got the ratio of the concentration inside versus outside, inside divided by outside. And so it's basically and like I said, the way to think about it is just imagine you start off at neutral, start off at 0, and then you have a channel that lets an ion through. It could be a negative or a positive ion, and that negative or positive ion will diffuse in whatever the direction is. Where there's a higher concentration, it's going to diffuse toward the lower concentration. And so the Nernst potential for a particular ion is basically just defined by what the concentration ratio is of that ion. So I'll always have a question like, say you have a negative ion, and there's more of it inside the cell. What's the Nernst potential? Is it positive or negative? That's a simple example. And what's the answer there? Well, if there's more of it inside the cell, brush that out of your mind and say, we'll start at neutral. We've got more inside the cell, but everything's completely neutral. And then some of it, since there's more of it inside, wants to diffuse out. And so the inside, you'll take away negative from the inside. And what does that do? That makes the inside positive. So even though there's more of it inside, the Nernst potential for it will be positive because it's trying to diffuse out. So that's the Nernst potential question. There's always one of those. And then we talked about the action potential. And that was caused by voltage-gated channels. And the voltage-gated channels, these are channels so the Nernst channels are just always open. They're just jammed open. And remember that even though they're always open, it reaches an asymptote. It reaches sort of like a fixed Nernst potential, where the same number of ones are going in as going out. And what's the reason for that? Well, they're just, if some positives went out and made the inside negative, well, the inside's negative, it's kind of attractive to positives. And so they'll at some point, they'll eventually be attracted back in. So it's a balance. But when we talk about voltage-gated channels, now we've got gating, and gating sort of means opening and closing. And so when we look at the gating part, we divided it up into two things. There was, activation, that was like opening the channel. And then if we stop doing whatever we did that opened the channel, there's deactivation. But there's this separate process, this separate active process of closing the channel that's also voltage gated called inactivation. And that's a sort of a counterintuitive thing to think about. And the idea is that if you depolarize the cell, the channel becomes activated, meaning it starts to open. But with a time delay, it's got this other process going on, which is the inactivation, also voltage gated. So what happens is you depolarize the cell, you'll open the channel and activate it, but at the same time you'll trigger this inactivation process, which will close the channel. So it's more complicated because of the interplay of these two processes, and that's pretty much what causes the spike to go in one direction, because there's a wave of channels behind the spike that are all inactivated, and so the spike can't go backwards. So that was the action potential, and we talked a little bit about there's a lot of these voltage-gated channels on the dendrites also, and they can sort of modulate the response of a cell. So then we talked about the postsynaptic potential. And the postsynaptic potential, it always looks like all graphs in neuroscience look like that. It looks like that. And so what causes this? This is caused by ligand-gated channels or neurotransmitter-gated channels. So these are channels that open up when a little molecule like glutamate or something sticks to them, and then it pops the channel open. And then the channel eventually closes because the glutamate diffuses out of the way, or some glutamate picker-uppers kind of like grab it out of there. And so eventually the channel closes. And so these, we talked about four main ones. There were the AMPA channels and NMDA channels and GABA A channel, GABA B channel. There's more than that, but those are four biggies. So those are, and those occur at synapses, and that's how one cell signals another. And then we talked about, in more detail, about the NMDA channel and long-term potentiation, LTP, and spike, timing, dependent plasticity. And so the idea there was these are both. The NMDA channels, are voltage plus neurotransmitter. So both of those things are required. It's an AND gate. Requires two things to open up. And so those NMDA channels, if you have both of those conditions, then the neurotransmitter means that the presynaptic cell was active, and the voltage depolarization means that the postsynaptic cell was active. Then the channel will open, let in some calcium, and then a whole bunch of stuff happens, and then the synapse gets stronger. And then we talked about spike timing-dependent plasticity, which was that if you just got stronger, everybody would just eventually blow up. And so spike timing-dependent plasticity turns out if the spike comes in just a little too late to help out, it gets punished. If it comes in just before the spike occurs in the second cell, then that's good. And if the time offset is more than about 1/10 of a second, nothing happens. So that was the spike timing dependent plasticity. And this is basically how we could do, a Heavian learning, which means correlation dependent, I-O, correlation dependent learning. So then we talked about dendrites. This was kind of a somewhat confusing lecture, but we made a little model of a dendrite. How does the current actually flow around in a dendrite? So to a first approximation, if we just imagine the outside of the dendrite is just a wire, which means everything's connected up to everything else, no resistance, the current can flow wherever it wants. we can sort of think of each little chunk of the dendrite as each little chunk of the membrane of the dendrite as a capacitor and a resistor in parallel. Now, that's just our conceptual model. The actual dendrite is just a thin layer, lipid bilayer. But that lipid bilayer has two properties. One, it has some capacitance, which is an ability to kind of get charges stuck to it of opposite signs across it. That's the capacitance. But then separately, it has a resistance to current flow, so it doesn't allow very much current to flow out. Then if we go inside the cell, there's some goo inside the cell, but that's more conductive. And then here's another little tiny, conceptual chunk of the membrane that has a capacitor and resistor, and it's hooked up to the outside. And so the problem was, trying to understand how this works when something is changing, like if we're injecting. So if we inject some current here, so here's a little electrode. If we inject some current into the inside of the cell there, how does this work? Well, if the capacitors are not charged, they're going to suck up some of the charges temporarily until they kind of get filled up. And because of that, there's a little time course. So you kind of have to fill up as soon as they start getting a little filled up, they get some effective resistance, and then more current can flow down. But then there's other capacitors down there that have to be charged up. But eventually, if you just turn the current on like a step, you reach a steady state. And then if you turn the current off at that point, all the charges that are stuck to the capacitors will belch out of the system, and you'll see so it's kind of like this goo. You push on the neuron, and then it's sort of charged at the capacitors. Everything sort of gums up the works a little bit, and then if you stop pushing, suddenly some current comes out. And so that accounts for why when you stimulate a dendrite, it takes a little while for the effect to propagate down. In real life, it's actually propagating down instantly, but most of the current is being sucked up by the capacitors right where you're stimulating. And then you have to sort of add that whole mess into the voltage-gated channels, because the voltage-gated channels are all dependent on exactly how much current got stuck into the capacitors, to determine whether they open or not. So you can see, it's a little complicated, but this basically, will cause, a delay, a dendritic delay in the effect of a synapse, like a postsynaptic potential on a dendrite. So then we talked about these very simple models. And the first one was a very simple Heavian learning model. And the idea here was maybe this is how we could make simple and complex cells. So that was kind of like the bottom line of this Heavian learning model. But what was the model? Well, the model was just ridiculously simple. The model just said the change in the weight is just equal to the input times the output. And that's an example of a correlation. So if you multiply two variables times each other and add them all up or average them, that's the definition of a correlation. And so when you see input times output, what that means is you're changing the weight proportional to the correlation between these two variables, input and output. And that's kind of basically what an NMDA channel does. But then when we're thinking about this conceptually, we know what the input is. That's just one input. But what is the output? Well, the output is the sum over all the other inputs times their weights. And so we can sort of boil this Hebb rule down to just inputs. That was the important point. And so what is this? Well, this is kind of the correlation, the correlation with every other input. So that gives us an idea of how does a Hebb rule work. Well, if you've got if you have a neuron and it's got all these inputs, it kind of looks out at its inputs, and it finds which ones are correlated with each other. And the ones that are correlated with each other, their weights go up. And so it makes the neuron, a Heavian learning rule will make the neuron more sensitive to whatever it's seeing. So this is a dumb rule. It's unsupervised. Nobody's telling it what the right answer is. It just makes the neuron more sensitive to whatever the range of things that it's seeing. So that was Heavian learning. And one of the ideas was that maybe this is how you can create those little rows of cells for the Hubel and Wiesel simple cells. So, there's several stages. One of the stages is the neurons that come from the center of the receptive field, the weights going to the neurons in the center of the receptive field have a little bit of a leg up. So, if you imagine there's a bunch of cells, and they're all going to this guy. They're all projecting to this guy. But as you go out into the periphery, less of them project. These guys don't project. But when we consider what the correlation is, if we've got some local correlations here, and we've got some local correlations here, these guys are correlated with each other, and they connect. And so these weights get a little bit of a leg up. These guys are correlated with each other, but only one of them connects. And so there's going to be an advantage for the ones in the center. So, and then if you go to a higher layer, there's some advantage for lining up the inputs in rows. That results in a slightly more correlated situation. And so that's how you might get something like a simple cell without any genetics instructions for it. That's really what the point was. This may be how the nervous system self-assembles. How does it sort of create something like orientation selectivity without having to rely on like genes to wire up those little lines? So that was Heavian learning, very simple model. And then we talked about, now that was just all feed forward. So there's no feedback at all. There's no time really. You just have you present a stimulus and then you change the weights, and that's it. There's no activity circulating around. But then we talked about attractor networks. And so attractor networks were kind of the polar opposite. Everything's connected up to everything, and the weights don't change. So this was, so feed forward, change weights. And then the attractor network was, all feedback, everything's connected up to everything, fixed weights. Now, we said you can have an attractor network where the weights are changing, and you can imagine that's going to be more complicated. But if you just look at the simple attractor networks, I always have a little problem about the attractor networks, and I'll always describe how to update the network. But the simplest kind of attractor network is you've got these thermometer neurons, and since everybody connects up to everybody, we'll take the output from each guy and connect them up to all the other ones. And so that means 9 possible connections if you include connecting up to yourself, which sometimes cells do. And so there's 9 possible weights here. So these guys were the weights. And there's going to be, there's an input, and then here's an output coming out of that guy, which is the same as an input going into this guy. So how do these things behave? Well, they have this tendency to kind of get stuck in a particular firing pattern. And so we could distinguish stable states. So we had the concept of a state. And what is a state? A state is just like some setting of all the activation values of all the neurons in the network. So it might be, like a state might be, say, +1 for this guy, +1 for this guy, and say -1 for this guy. So that's an example of a state. It means just sort of like a condition of the network. And so we have a state, and then we have stable states, And what's the definition of a stable state? That's just a state that when you update any unit, nothing changes. That's the definition of a stable state. You can sort of get stuck there. And what's the definition of an unstable state? That's any state that when you update any of the units, a change occurs. And what was the behavior of these neurons, of these networks? Well, they sort of tend to go to an attractor. So, if you started off here, it will say, go to, say this guy is an attractor right here. And there's another attractor here. So if you start off here, it goes there. Start off here, it goes there. If you're just on the other side, it goes there. So the idea was that the example that we had was more like the corners of the cube. So say we start off, here's the back of the cube. So some of those corners of the cube are going to be stable and other ones are not. So say we start off in this guy. Say this guy is not a stable one here. And then what will happen, you update it, and maybe it goes to this guy. And let's say that corner is a stable one. And that one's a stable one, then if you update everybody, nobody changes. And there might be some other unstable ones. So all these guys are unstable ones, and they just sort of head toward the corner, the closest corner. It's kind of the same thing. It's about the same thing as this. So that's a typical way that neurons can behave, and all you require for this is that, in general, the weights going one direction are similar to the weights coming back. So we said that was an example of a symmetric matrix of connections. So what does that mean? Like, here's the diagonal of that matrix, and a symmetric connection means these two are similar. So what are those two weights? Well, that's the weight of this first one. You can see is the weight of this unit going to unit number 2, and then what's this weight? This weight is unit 2 projecting back to unit 1. So if the connections from a unit are similar like that, then you get these attractor states. But it turns out they don't even have to be similar. They can be quite different, and you'll still get them. If they're exactly the same, you're guaranteed to have attractors. And so this was just a way of sort of thinking about how giant globs of neurons all connected together behave. This is like a real typical way they behave. You start them off someplace, and they jiggle around for a while, and then they sort of get stuck into a particular pattern. Is that a good model for the brain? No. Because clearly, if I look at my marker, I don't go marker and then just get stuck there. Obviously, that's not the way the brain works. But this is just very simple ideas, sort of of ways of thinking about how the brain works. And I'll have problems on this. There's here's the update rule. Update this network. Is it stable? Is it in a stable state? Is it in a stable state after you updated it? Or what is the energy of the network? And you remember, the energy of the network was you just go to each weight and look at you basically sum across all the inputs times every weight times every output. That's just a single number that sort of says something about the network. So you just go to each of the 9 weights, multiply the input times the weight times the output, sum the whole mess up, take the negative of it, and then you have the energy. It's kind of like, the idea is based like on physics, where if you look at this complicated situation, like a car at the top of a hill, and then a car at the bottom of the hill, and you can say, like, there's a certain energy to that. There's some potential energy, it could fall down, and there's maybe the car is already moving, there's some of that energy, and we add them all together and we get one number. But that number is helpful for kind of understanding how, that the car is going to continue downhill. So that's kind of what it was based on. Okay, so then we talked about development. I usually don't ask real specific questions about development, but I make you reproduce this all-important diagram of how the brain is basically set up. You got the 4th ventricle there, cerebellum's over the top. It's the rule of Serino because there's the pons midbrain junction where everything flips, and then you got the midbrain and the thalamus, and then the forebrain sticking out like that. And the ventricle closes up again right there, and here's the ventricle down in the spinal cord. And then it opens back up again here, just a little opening, and then in pockets to form the two hemispheres. And so I always want you to reproduce this diagram. It's a basic sort of diagram of the brain. And you have the forebrain here, and the thalamus, and the midbrain, and then you get the pons midbrain junction. You got the cerebellum here, which is behind the pons midbrain junction. And then we got the pons, and then the medulla, and the spinal cord. So this is a good thing to keep in mind because it's just the basic layout of the cylindrical coordinate system of the brain, the neural tube, the original neural tube. So then, now we're sort of like into recent history. So then we talked about the retina. And so we had, the basic setup of the retina. And, you start off with the photoreceptors. And then we get, bipolar cells. That's how you get the on and off. And then you got ganglion cells. And there was a whole bunch of these guys. So this is where the on and off comes from. But then in the ganglion cells, you had, the X-like ones and the Y-like ones and the cone, the blue ones. And then we get up to the cortex. And we talked now we're sort of like stuff in recent memory. So we talked about how do these streams work. So the streams start in the retina, but then they keep going on. They keep splitting up, and then mixing them together, and then you get new streams. And this was just sort of introducing the concepts of how we've got like one stream of information, but then we're somehow making multiple copies of it that emphasize different things. So, the example was blob cells. Like, why would they throw away orientation? It's hard to construct orientation. Why would they actively throw it away? And the idea is maybe they're trying to just detect how bright something is without getting confused about whether there's an edge there. But then, equivalently, the orientation selective cells really don't care about brightness as much, and so they're going to throw out some information about brightness because they really want to tell you, like, what is the orientation of this edge? And I don't really care if it's like a really bright edge or it's just like a dim edge, or maybe it's got black on one side of the edge or vice versa. So that's one of the things that the streams do. They kind of toss information of a certain kind and focus on information of a particular kind. And that's a theme that it's done hundreds of times throughout the brain. So it's a real basic organizational principle of how the brain works. And that brought us up to simple and complex. So any questions about the material? Yeah. Oh, the rule of Serino. Yes, so of course I can explain the rule of Serino. So the rule of Serino basically is if you're in the body, so here's the body out here. So if you're in the body and you connect up to the brain, if you're behind that junction, you'll just go into the same side. So what's an example? For example, the ear. So, the ear, the left ear projects into the it actually goes to the up to the pons right there. So it goes up to the pons, but it goes up to the left half of the pons. Now, if you're in the body, part of the body that's past there, like the eye, for example, the eye the eye is going to project to the opposite midbrain and the opposite thalamus. So that's the first part of the rule. That's like the body part of the rule. But the other part of the rule is that if you're in the brain, if you say you were in the forebrain, and the forebrain is going to project to a cell in the spinal cord, in that case, it's going to go down. Now, it won't cross immediately when it passes the pons midbrain junction. This one waits a little while, but it won't touch anybody. It won't synapse on anybody until it gets down here, and then it will synapse on the spinal cord. And then, that will go down the spinal cord. And so that's, if you cross the junction, you're going to cross. If you stay on either side of the junction, connect to somebody on the same side of the junction as you, then you won't cross. So if I'm looking at the midbrain, and the midbrain projects up to the thalamus, say the superior colliculus goes to the pulvinar, that's an example, that's going to be that will be 100% uncrossed, because they're both past that junction. So it's just like, it's basically, we just flip the left-right specification of the brain at the junction, at the pons midbrain junction. Okay, any other questions about whirlwind tour of lower level neurophysiology? We'll slowly work ourselves up to sort of more conceptual things as time goes on.